Hi everybody, in this video we are going to finish up biochemistry. We're going to go through the carbohydrates and the lipids. This might be a slightly longer video, but it's not too many slides. So first we're going to look at carbohydrates. Carbohydrates are sugars. They are the molecules used as energy or storage in mammals and plants. All carbohydrates contain either an aldehyde or a ketone group. If you have an aldehyde group, you are called an aldose. So aldoses contain the aldehyde group. You can see the carbonyl group at the top carbon. That's at the end of the chain. If you have a ketone group, you're called a ketose. So ketoses contain the ketone group. You can see on the right, that's the ketose. The carbonyl group is on the second carbon from the top, so it's in the middle of the chain. They will have either an aldehyde or a ketone, and they all have an alcohol functioning group. So they'll have at least one, oftentimes multiple. So it's carbohydrates that we make ethyl alcohol from. That's what we drink. And it's because when you break down, you ferment the carbohydrate, out comes these little alcohol groups. So these little hydroxyl groups. So we can categorize carbohydrates by whether or not they're simple or kind of compound. These simple sugars are the monosaccharides. Then you have disaccharides, which are two sugars, and then polysaccharides, which is more than two sugars. So the monosaccharides are the simplest of the sugars, and they cannot be hydrolyzed into any simpler sugar than that. So monosaccharides cannot go through hydrolysis. We name monosaccharides by the number of carbons, and then we put an ose at the end. So if you look at these, if you have 3 carbons, you're a triose. If you have 4 carbons, you're a tetrose. If you have 5 carbons, it's a pentose. If you have 6 carbons, you're a hexose. And if you're a 7 carbon, you're a heptose. The most important ones that we're going to look at are the pentoses and the hexoses. So the pentoses, the 5 carbon monosaccharides that are important to us, are mainly important in our DNA and RNA because there are 2 pentoses. They are ribose and 2-deoxyribose. They are both the sugar portions of nucleic acids, so DNA and RNA. For the ribose, if you hydrolyze ribonucleic acid, RNA, you will get ribose out of it. Again, that's why it's called RNA, ribonucleic acid. If you hydrolyze DNA or deoxyribonucleic acid, you'll get 2-deoxyribose. Now, if you notice, ribose and 2-deoxyribose are basically the same structure, except if you go to the second carbon down on the 2-deoxyribose, you'll notice that it's missing an oxygen. That's why they call it 2-deoxyribose. Yep. So these are important in our DNA functioning. The hexoses, or the 6 carbon monosaccharides, are important to us in the realm of nutrition. So they're the most important nutritional carbohydrates. They all have the same molecular formula, C6H12O6, but there are about 24 isomeric variations in between them. So again, an isomer has the same molecular formula, different structural formula, so I can take these components and mix them together to get different hexoses. So there are 2 important hexoses we're going to look at. One of them is glucose. Glucose is blood sugar. You can also call it dextrose or grape sugar because it's the same sugar that you find in grapes. We store glucose in our liver and our muscles as glycogen. If you remember, we've seen glycogen before in decomposition. Glycogen is animal starch. It is a polysaccharide that you store in the muscles and in the liver, and when you need it, it gets broken down, it gets oxidized back into glucose. Insulin is what controls the storage of this glycogen, and so lack of insulin production causes increases in blood sugar. Now, there's two kind of conditions you can have in relation to glucose. You can have hypoglycemia, which is low blood sugar. People with hypoglycemia oftentimes have to eat a lot because otherwise their blood sugar gets low and they get kind of lethargic. More dangerous would be hyperglycemia. Hyperglycemia is high blood sugar. This would be like diabetes mellitus. This can lead to circulatory problems. Usually, when you have problems with diabetes mellitus, it's because of poor diet and exercise. And so you're eating a lot of sugar, but also a lot of fat. And so when you are hyperglycemic, this blood sugar will not get stored in your body properly. And so if you eat a lot of sugar, your body's not metabolizing it properly. It's going to actually start to metabolize fat a lot more, and you get a lot of lipids floating around in your system, which leads to arteriosclerosis, and eventually they start cutting parts of your body off, mainly starting at your feet and working their way up. So hyperglycemia is much more dangerous. The other important hexose we look at is fructose. Fructose is also called levulose or fruit sugar. So this is the sugar that you would find in most fruits. It is found in fruits, honey, and semen. It's actually the energy source for semen, so it can swim. It is also the sweetest sugar. If you've ever heard of high fructose corn syrup, well, it's just fructose that they get out of corn. It's very cheap and very sweet, and that's why it's used as a sweetener in a lot of foods. So those are the two main monosaccharides we look at: glucose and fructose, blood sugar and fruit sugar. The disaccharides are made up of two monosaccharides. So if I took a disaccharide and put it through the process of hydrolysis, then I would get two monosaccharides out of it. So here you can see I take two monosaccharides, I hook them together, and I get a disaccharide. And out will come a water molecule. So that's a condensation reaction. The molecular formula for disaccharides are all C12H22O11. They're all pretty much the same molecular formula. Again, they just have different structural formulas. So there are two main disaccharides that we look at. One is lactose. Lactose is milk sugar. This is made up of glucose and galactose. Glucose, again, is blood sugar. Galactose is brain sugar. And this is synthesized in the mammary glands of mammals. So when you have milk, and if you let the milk sit out for too long, the lactose in the milk can get turned into lactic acid by lactobacillus bacteria. And this is what causes sour milk. So if you have milk that's soured, the lactobacillic bacteria has gotten in there and converted the lactose to lactic acid. Now, some people can become lactose intolerant. Lactose intolerant means that you have a lack of lactase. Lactase is an enzyme. It's the hydrolytic enzyme for lactose. It breaks lactose down in your intestines, and so you can absorb it better. All humans, or most humans, as babies, have high levels of lactase. That's why they can ingest milk. That's what they live off of. Very few babies are lactose intolerant. But as you age, you tend to not produce as much lactase. And that's why most people who are lactose intolerant are older. Once the lactase stops being produced, you can't break down the lactose, you can't absorb it into your bloodstream and through your intestines, and you start to get inflammatory responses to these sugars. Sugar in general tends to be an inflammatory substance, and so that will inflame your intestines. You'll get stomach cramps and a lot of diarrhea. So if you're lactose intolerant, stay away from the cheese and the ice cream. The other disaccharide we're going to look at is sucrose. Sucrose is table sugar. It's what comes out of sugarcane. It's made up of both fructose and glucose. So glucose, again, is blood sugar. Fructose is fruit sugar, and it's the sweetest, and that's why table sugar is very sweet, because it contains fructose, the sweetest of the monosaccharides. So those are the two disaccharides we look at, lactose and sucrose. Next, we're going to look at polysaccharides. Now, polysaccharides are made up of many monosaccharides, and so technically, if you put a polysaccharide through hydrolysis, it will actually create a bunch of disaccharides, which will then get hydrolyzed down into a bunch of monosaccharides. Remember, monosaccharides cannot go through hydrolysis. The important polysaccharides are things like starch. That's how plants store sugars. So like potatoes are very high in starch. Glycogen for us is animal starch. That's how we store glycogen or store sugars in our body. Another really important polysaccharide is cellulose. Cellulose is the main component in plant cell walls. It's basically wood is cellulose. We use cellulose in embalming for cavity powders as a way to suck up moisture, but also as kind of a filler. The interesting thing about cellulose is that it is the most abundant organic compound in the biosphere. The biosphere is where living things are, and so there's more cellulose than anything else. And so those are the different classifications of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Now, if we look at how carbohydrates get broken down, they can get broken down through hydrolysis. They can get broken down through oxidation, which is actually how we break them down for energy inside the body, and they can be broken down through fermentation. So the hydrolytic breakdown of carbohydrates, or saccharolysis, if you will, if you remember, monosaccharides do not go through hydrolysis. You can't hydrolyze a monosaccharide. You can oxidate a monosaccharide. You can ferment a monosaccharide, but you can't make it go through hydrolysis. The disaccharides can go through hydrolysis. They get broken down into two monosaccharides. And the polysaccharides can also be hydrolyzed. They get hydrolyzed down into disaccharides. The disaccharides then get hydrolyzed down into monosaccharides. So if you remember, hydrolysis is just the process of breaking down polymers. Well, that's all polysaccharides and disaccharides are. They're just polymers of monosaccharides. So once these polymers of monosaccharides get hydrolyzed down, those monosaccharides can go through other types of breakdown. One of those is oxidation. This is how we break down monosaccharides in our bodies. Oxygen is used to break down glycogen, and that will then turn into glucose. So that's produced. This process of oxidation will produce CO2, water, and energy. 1 gram of sugar will produce about 4 kilocalories of heat. And when they say heat, they just mean energy. The interesting thing about that is when we get to lipids, you'll see that lipids actually will make about 9 kilocalories of heat. So you actually get more energy out of lipids than you do out of sugars. Fermentation is the enzymatic decomposition of monosaccharides. And so mainly hexoses get broken down through fermentation. It will take an enzyme to go through this. Zymase is a very common enzyme for fermentation. But what comes out of fermentation is ethyl alcohol and carbon dioxide. Again, fermentation takes place in the body when the body dies, and the CO2, the carbon dioxide, is the main component of tissue gas, the gas formation that is one of the signs of decomposition. This is performed by aerobic bacteria, and so if you want to make some moonshine, you take sugar, usually sucrose, put that in and start to boil it. It starts to break it down. The bacteria will come in and ferment it, and out comes ethyl alcohol, moonshine. Now, where carbohydrates really come into effect in embalming is in rigor mortis. And the reason that they affect rigor mortis is that rigor mortis is caused by a lack of ATP. ATP is synthesized from glucose. Okay, so Technically, it's all based on the lack of oxygen, but when the body lacks oxygen, it can't make ATP, and ATP is what makes your muscles relax. So ATP stands for adenosine triphosphate. It is the body's main energy carrier. It gets made through glycolysis. It gets made through the Krebs cycle. It also gets made through the electron transport chain. So there's various mechanisms that we're not going to go through that create ATP in the body, in the cells. Okay. Again, it's synthesized from glucose through all those processes. So glucose, again, is a carbohydrate, and as long as we have glucose and oxygen, we can make ATP. Where it starts to affect rigor mortis is that when the muscles contract, anytime your muscles contract, that's called the actin-myosin complex. And essentially, this is what rigor mortis is. Remember, it's just a full body cramp. They describe your muscles as having fibers. They have the actin fibers, those are the thin muscle filaments, and the myosin fibers, which are the thick muscle filaments. And when you have muscle movement, the actin-myosin complex is the contraction of the muscle. To make your muscle release or relax, that is the ATP-myosin complex. That's relaxation. So as long as your body's making ATP, your muscles can be relaxed. When it stops making ATP, runs out of oxygen, that's when the actin-myosin complex starts to form again, and that's where you get rigor mortis from. That's why bodies don't tend to go through rigor mortis right away. They go through primary flaccidity first because your body's still making ATP and the actin-myosin complex is undone. When you get to the rigor complex, which again happens about 2 to 4 hours to form, roughly, that's when the actin-myosin complex can no longer be undone. You're not making ATP because you don't have any oxygen. Now, I've mentioned this one before, but I think this is the first time it shows up in the notes, is instant rigor. Instant rigor is when rigor sets in immediately, and it's usually caused by, as you die, you're tensing your muscles. You're tensing your muscles, and it's burning all of the oxygen out of your system. Usually this happens in cases of traumatic death. Again, it's anytime you're almost getting in a crash, you kind of tense all your muscles up. That burns out the oxygen supply in your system. That means there's no oxygen left to make ATP. The muscles automatically go into rigor. So you're not going to see it very often, but in cases of car accidents, any kind of traumatic death, you might see instant rigor. So that finishes it up for carbohydrates. Next, we're going to look at lipids. Lipids are just fats, okay? But there's lots of different types of fats. Generally, though, they all have very similar characteristics. Most fats are insoluble in water. Fat is hydrophobic. It doesn't like water. And so they don't mix very well. This is kind of exemplified in Italian dressing. Most Italian dressing you have to shake up because you have to make the water and you have to make the oils in the Italian dressing turn into little globules that will kind of disperse throughout the water. They're not mixing though. They're just being suspended. I can get insoluble fats to mix with water if I use what's called an emulsifier. So emulsification is the act of mixing two insoluble liquids. And there are certain chemicals that you can use as emulsifiers to get these to mix. Fats are found a lot in the brain and the nervous system. You find a lot of fat covering the myelin sheath, in the myelin sheath of nervous cells. Again, brains have lots of fat in them. They are the structural component of cell membranes. Your cell membranes have fats in them, and we also use them as energy storage. Now I can classify lipids. I have simple lipids, and I can have compound lipids. The simple lipids can go through hydrolysis, and they get turned into fatty acids and alcohol. If you remember, fatty acids are monocarboxylic acids. So when you hydrolyze simple lipids, you get fatty acids and alcohol. The simple lipids you can categorize as fats and oils and waxes. So Fats and oils tend to get hydrolyzed into fatty acids and glycerol. So fatty acids, again, are monocarboxylic acids. Glycerol or glycerin is a trihydroxy alcohol. Fats are considered simple lipids that are solid or semi-solid at room temp. Oils are simple lipids that are liquid at room temp. That's kind of the difference between them. There's also essential fatty acids. Essential fatty acids are fatty acids that the body cannot synthesize. They are something that has to be part of the diet for you to be healthy. Again, fatty acids are the monocarboxylic acids. You also have waxes. Now, waxes are esters of fatty acids and heavy alcohols. If you remember, we talked about esters in chemistry. Esters are made through the combination of alcohols and carboxylic acids. The waxes that you tend to run into are things like lanolin. That is a wax found in sheep's wool. Lanolin is very commonly used as a humectant in embalming, but lanolin should not be used as an internal humectant. Lanolin is a colloid, and if you pump that into the body using it as a humectant, what will actually happen is, because it's a colloid, it'll get into the capillaries but not get through the capillary semi-permeable membrane because it's a colloid. It will sit inside the capillaries and not diffuse. And what that can actually do is create a hypertonic environment inside the capillaries. And so you're pumping lanolin into the body to help to moisturize the tissues, help it retain moisture as a humectant. What it's actually going to do is dehydrate the body because it's sitting inside the capillaries, making a hypertonic environment inside the capillaries. That's going to suck moisture out of the surrounding tissues. So you don't want to use lanolin internally as a humectant. It's a very good humectant externally, so on skin and things like that. Beeswax, that is a wax. That's what we use. That's the main component of most of our restorative waxes. There's also an interesting one called carnauba wax, or more commonly known as sex wax. That's what we use on surfboards and skis as a lubricant. So whereas the simple lipids go through hydrolysis and turn into fatty acids and an alcohol, compound lipids will hydrolyze down into fatty acids and alcohol, but other stuff. They have other components in them. One of the common compound lipids are phospholipids. Now, you find phospholipids in the body as part of the structure of the body too. But phospholipids consist of glycerol, that's the alcohol, 2 fatty acids, the monocarboxylic acids, 1 phosphoric acid, and 1 amino alcohol. Now, that amino alcohol is kind of important to us because when we talked about formaldehyde and glutaraldehyde only interact with proteins for the most part. But there are some phospholipids in the body that could get cross-linked with formaldehyde and glutaraldehyde. Now, phospholipids are often used in foods, in the production of foods, because phospholipids will act as emulsifying agents. Again, these are things that we could use to mix 2 insoluble liquids together. So if we look at the reactions of lipids, the breakdown of lipids that they can go through, in your body you can metabolize lipids through oxidation, same process as breaking down sugars basically. So you use oxygen to break it down. 1 gram of lipid will produce CO2, water, and 9 kilocalories of energy. Again, if you remember that carbohydrates only produce 4 kilocalories of energy. And that's why when you look at like survival shows, oftentimes they're looking for things that have high lipid content, high fat content to eat because you get more energy out of it. This is the basis of the ketogenic diet also. So fats have higher caloric value than carbohydrates do. Okay? The body will burn carbohydrates if they're there, but if you get rid of carbohydrates, then it will start to burn fat. So that's how we metabolize fats. We talked about saponification in embalming. Saponification is technically described as the boiling of lipids in an aqueous alkali, but Through that boiling, you're actually hydrolyzing those lipids, again, under an alkaline condition. The products of saponification are basically glycerol, that trihydroxy alcohol, and a soap. Again, this is how they used to make soaps back in the day, is they would boil animal fat under alkaline conditions and get this soap. And soaps are just metallic salts of fatty acids. The soap that gets produced through saponification is adipose sear or grave wax. That's the saponification of the body's fats. And most of the time, you're not going to see this unless you bury the body in alkaline soil. So you put a body in, let's say, a wooden casket and in a grave liner, the water will be able to get in, and if it's alkaline, that can start this saponification process. The other type of breakdown is rancidity. Rancidity is exposure of lipids to the air. And technically, there are two types of rancidity. There's hydrolytic rancidity. That's the microbial hydrolysis of lipids. So microbes can get onto the fat. They will start to hydrolytically break down the lipids through their enzymes. And this is what creates the foul-smelling organic acids. The hydrolytic rancidity can be one of the things that contributes to the odor of decomposition. The main odor from decomposition, as we know, are ptomaines, the indole, scatole, putrescine, cadaverine, but you could also get some foul-smelling organic acids in there. You could also get some of the thiols or mercaptans smelling in there also. Oxidative rancidity is when it gets exposed to air. So the oxygen in the air oxidizes the carbon-carbon double bonds in the lipids. This will create foul-tasting aldehydes and ketones. And that's why one of the reasons why you get a lot of antioxidants added to food to prevent oxidative rancidity, because you don't want it to taste bad. We also use antioxidants in the body to help protect the brain, essentially, from the actions of oxidation and oxygen also. So that finishes it up for lipids and biochemistry. The next video, we're going to start looking at the process of how formaldehyde and glutaraldehyde fixate proteins.